Welcome to Decoded, a podcast about how data, technology, and AI are transforming healthcare. I'm Yogi Kamblong, a biologist, entrepreneur, and dreamer, and the co-founder and CEO of Sophia Genetics. This series is about more than science. It's about the people using data to close the gap between what healthcare could be and what it must become. It's about equity, truth, and the power of interpretation. Because data alone doesn't save lives. People do. My first guest today is Professor Umberto Malapelle, someone who has never lost sight of that purpose. If you're in oncology or diagnostics, you already know his influence. He leads the Predictive Molecular Pathology Lab at the University of Naples. He's editor-in-chief of the Journal of Liquid Biopsy, scientific secretary of the International Society of Liquid Biopsy. But beyond the titles, he's a global voice for diagnostics, precision, and access. He's helped shape how we understand diseases, how we read signals from tissue and blood, and how we bring those insights to every patient, not just a privileged few. He believes, as I do, that the future of healthcare must be intelligent and fair. Let's dive in. Good afternoon, Umberto.
Good afternoon. Thank you very much for this nice introduction. Thank you.
You're welcome. So most of us, Umberto, have been already impacted directly or indirectly by cancer. But while most of the people know what an oncologist does, few know what is the job of a pathologist and the importance of that role in oncology. So could you tell us what pathology is about to start?
So it's the backstage. For this, the people already don't know the role because we are working all together in the backstage of the anatomic pathology department with several peoples within the anatomical pathology department, because you have pathologists, biologists, biotechnologists, technicians. So it's a sort of multidisciplinary teams that works all together with the aim to decodify the complexity of biometrics that we receive in the labs, so tissue and liquid biopsy. And it's a really fascinating role in this time that we are living. It's really complex, but for sure we know that this is the way that we need to follow to have all the information before to start with any type of treatment selection.
And so in the context of a patient journey, you can take the cancer you believe is the most pertinent. When a pathologist will be engaged in the workflow of the patient to support a decision around treatment?
More than one time, because at the beginning, you need to make the diagnosis. And then after the diagnosis, you need to characterize several types of prognostic biomarkers. Then in the middle, because you need to define the therapeutical strategies for your patients. And so another time you need to characterize other type of biomarkers, so-called predictive biomarkers. And then at the end of the process, because you need to decodify the complexity of this type of characterization with the oncologist and the other people that are following the patients. And then also in dynamic facet, because several times you need not only to characterize, but also to recharacterize the mutational status of the patients during the path.
Taking a big step back at the very basic levels, you have a patient, right, who has cancer. And then most of the time, I don't know whether it is always in the context of the surgery, but there is a piece of tissue that is being taken, right? And so I guess this is where pathology starts, if I'm not mistaken.
Sure, absolutely. You are right. And this is in a step before the surgical resection, if you can, because you need to take into account that you can have two different types of patients for which it's possible to proceed with a surgical approach, or patients for which it's not possible to proceed with this type of approach, and you need to proceed with the other type of approach, like as pharmacological treatments. In this case, you proceed to have a diagnosis or a small piece of tissue that is called or biopsy or cytological samples. And another time you have the anatomic pathology department that are supporting this type of activity.
Okay. So the tissue in most of the cases is taken as the patient gets the surgery. Then I understand there are some, bits of the tissue that goes to the pathology lab. And long time ago, pathologists were looking only at the tissue under microscope, right? Can you explain us before we go into the molecular aspects and how it is transforming pathology, what people were doing 20 years ago?
In past, the the end of the history is represented by a fully prepared section of tissue to be analyzed under a conventional optical microscope. And this is in past. But now you have the addiction starting from this basis, because the basis remain, that is the diagnosis. But starting from this basis, you can add some other strategies, like as an example, immunophenotypic evaluation of some biomarkers expressed by tissue, the so-called word of immunohistochemistry or immunocytochemistry, and then the molecular characterization, that is difference in relation to the phase in which you are working. An example, in the diagnosis is diagnostic molecular pathology, in the prediction is predictive molecular pathology, and altogether represents the new anatomic pathology department.
So pretty important department, pretty complex. Years ago was only tissue under microscope to see the shape of the cells and maybe some protein modifications that were known to be related to the cancer. And more recently, there have been the emergence of molecular pathology, right? So how transformative has been moving from tissue pathology first to molecular pathology, and why the molecular information is important in cancer?
So it's one of the revolution that we are living and that we have lived in past regarding the upgrade of these really fascinating disciplines, because it's the same history, but you are able to see the things from another point of view, because you are able to see not only the surface of the tissue that you are working for, but you are able to see also all the other things that are in backends and that explain why the tissue is in this case or in these other cases. So the keywords right now of the anatomic pathology department and the people that are working in this type of department is integration. You need to have the ability to integrate different sources of information.
Can you give us a sense of how molecular pathology has influenced how we're diagnosing and treating patients? Because my understanding is 20 years ago, nobody could sequence the DNA of the tissue of a patient, so nobody could really decipher the molecular information of a specific cancer. And today, this is being done quite regularly into the hospitals. And this has been kind of a revolution, right? So what are the type of mutations that today enable us to better act on cancer? Are we thinking about cancer today the way as we were thinking 20 years ago?
It's really simple to have an example in mind. In example, if you think to endometrial cancer, some years ago, the diagnosis is based only on the morphological parameters. But today, you need to be able to make a diagnosis to know also the molecular landscape. So, in example, if you are able to identify mutation in some gene like as POLE or macrosatellite instabilities or P53, you are able to identify in really precise way the diagnostic class. And so this is relevant not only to define the diagnosis, but also to set the prognosis for these patients. And 20 years ago, this is not possible, but now is a reality, and you need to have this type of strategies every day in your lab. And this is for the diagnostic part. From the predictive point of view, the scenario was completely changed by the introduction of predictive molecular pathology, because if you think that 20 years ago, in non-small cell lung cancer, we had for the first time the introduction of TGFR mutation-based analysis, and the patients have the opportunity to proceed with an pills and not with a chemotherapeutical-based approach like as in the past. And from 20 years right now, if you think an example in non-small cell lung cancer, we have more than 11 biomarkers registered in clinical practice that you need to analyze to proceed to select precise strategies for your patients.
And how quickly has this understanding evolved? I understand it's very dynamic, but so would you say that now, thanks to the sequencing of the DNA of the mutations, we better understand cancer by twofold, by tenfold? And how are we in our understanding of cancer overall?
So if you consider, in example, the introduction of our ability to make an access to next generation sequencing in clinical practice, we had then a really quantum leap. Because if you see the number of biomarkers approved after the introduction of next generation sequencing strategies in clinical trials is disruptive. Because in years 2018, more or less, when most of the part of the lab around the world have access to these type of technologies, we are able to co-characterize all these type of biomarkers in same time. And this is really relevant. years ago, this was not possible because, in example, when I starts these type of careers, the only biomarkers approved was KRAS mutation in colorectal cancer, and you need to cover only a small piece of DNA, and we in past used, in example, Sanger sequencing. But right now, it's not possible to proceed in this sense because you need to take into account the time in which you need to have this type of information. And if you need to analyze 10, 12, 20 different biomarkers for one tumors, for sure you need to have this ability in your hands. And this ability is explained by the power of this type of next generation approaches.
So move from tissue to molecular, you've been highlighted how many biomarkers have emerged that are now being associated to cancer and can be leveraged on by the pathologist, either to help the diagnosis or the treatment decision. You've been talking about technologies like Sanger sequencing and next generation DNA sequencing, which are technologies that enable us to decode the DNA of patients and basically understand what are the mutations. So more recently, the same principles now have been moving from tissue, which was the original, I would say, work of the pathologist, to liquid biopsy. Why is that? And what's the power of the liquid biopsy?
Because we need to be dynamic, not only static. And so by using biofluids like as bloods, you have this ability in your hands because you can collect during the time several times the source, in this case the biofluids from your patients, in really non-invasive way, because it's really more simple to have in your hands several times for the same patient's biofluids instead to tissue. And this is one of the first reasons. The second one is that you need to intercept some alteration in different times in your patients. In example, if we think to breast cancer, now we need to detect mutation after a resistance disease, after a line of treatments. So this means that we need to recollect the materials from our patients. And so liquid biopsy in this is really fast, really accessible for all the patients. And then by using the power of these type of technologies that we have discussed in liquid biopsy, another time you are able to see simultaneously not only one piece of the history, but the entire chapter that you are reading, because you are able to see all the landscape, all the mutational landscape in blood streams of your patients.
In liquid biopsy, it means you have probably material that came out of your tissue. And I've heard about cells. So for example, circulating tumor cell or circulating tumor DNA or other eventual biomarkers that go out and then circulate, as you said, into the blood and other fluids, right? Today, how a pathologist is leveraging on one versus the other, because one may think that, okay, a biopsy is not necessarily easy. It may require a surgery to be able to take a piece of tissue and decipher what is driving the cancer, while from blood looks like easier and more accessible. So can we replace tissue pathology, molecular testing by liquid biopsy, or are they complement? Can you explain us how these subtleties are emerging over time as we are understanding more and more the importance of molecular biology in cancer?
So there is not a war between tissue biopsy and liquid biopsy, but is a marriage, because you need to use one or another one in relation to the clinical need that you have. If you are asking to a liquid biopsy, which is your dream when you become fully validated, is not to substitute the tissue, but is to be in setting in which you are not able to have the tissue. So in example, early detection and cancer interception. So in the screening, large screening programs, and please, thanks to all the solid tumors, you now have the possibility to have biometrics to proceed with these type of screenings. But by using biofluids, for sure you can. But this means that you need to work on the analytical side to have in your hands technologies that are able to identify this type of alteration in really specific way, because you need to connect this type of alteration with the risk to develop in future and a tumor. So this is the ability of tissue and liquid biopsies to be complementary. And also in some other settings, like as advanced stage, if you think in the advanced stage, for sure, you need to have the diagnosis. So this means that you have the tissue. But if the tissue is not enough, because you need to carried out some other deeply analysis by using some other strategies like as immunohistochemistry and the tissue can finish. So for sure, you can complement your information by using biofluids. Another time, you can complement your information by using liquid biopsy. So in past, the people thinks that liquid biopsy need to replace tissue biopsy or vice versa. Is not the goal. As we have discussed before, the key words for the future is integration. So more information we have, better we can decodify all this complexity for our patients to design the path of the treatment. So isn't a problem not related to the biology side, but isn't a problem related to sustainability. Because the question is, can we in every patient analyze several biometrics by using several times next generation sequencing and the other molecular biology approaches? So we need to pay attention on this, if it's sustainable or not. But it's not a biological discussion because for sure, from pathologists, biologists, and all the other people that are working in lab, if we have more information, your diagnosis can be more precise.
Makes sense. And as you're talking about affordability, how much it costs roughly to do a proper molecular profiling of a cancer?
So the problem is not the cost of the reagents or the cost of a machine. The problem is the overall cost of the workflow. Because to make in place a test doesn't mean to buy a platform, but mean to have skilled people, to have a really deeply culture in management of this type of test, to have people fully trained to decodify the information coming from this type of the analysis, and then the reagents, the platforms, and all the other stuffs. So we have published 2 months ago and a worldwide analysis on cost comparison by using single gene testing strategies and multiplex strategies in setting of solid tumors. And we have defined that by using a maximized multi-gene testing approach, for sure you are able to reduce the cost. But in particular, the most important things is that by using the same time of your personnel, you are able to triplicate the quantity of the patients for which you can provide a service. So this is the main thing, is the people that are working in lab, is not the reagents.
I want people to know how much cost testing versus a treatment. Roughly, how much is it? 1,000, 1,500 euro?
If we compare the cost of the test respect to the cost of the treatments, the cost of the test is like as homeopathic.
Exactly, right? That's my point. So basically, it's about, let's say, 1,000, 1,500 euro to test properly someone one time versus a treatment of 50 to what, $80,000 a year, kind of?
Absolutely. Yeah, we can move in this range.
Exactly.
And then also if you need to repeat the testing during the time, this means that you need to change the treatment during the time. So our vision, and I'm saying our because it's not only my vision, but it's a vision of a group of people that working together every day, is like as all the predictive molecular pathology group that we have in Italy that are working on these points in Italy, but also the International Society of Liquid Biopsy community that works in this setting. Our vision is that the cost of the test is not a cost, is an investment, because it's the way that you are using to avoiding to use not performance treatments in patients for which you are not able to define the presence of a biomarker. So it's not a cost, because you are able to reduce the cost. If you are not able to having a test, this means that you need to proceed on the overall population with strategies, with therapeutical strategies. And we already know that this is not the path that we need to follow, because we can have not the same performance in all the patients. We can have some toxicity coming in patients in which you don't have this type of biomarkers. So this is the way in which you need to see the test. It's not a cost.
Yeah. Isn't this crazy? We all have cars, right? And we never get the car fixed before they do a diagnosis of what doesn't work anymore.
For sure.
How do you see this play in 5 years' time? Do you think that despite the complexity, we will have overcome these challenges on accessibility and we will have many more patients that will be properly diagnosed so that we can better understand their cancer, follow longitudinally with liquid biopsy?
So if we think in the past years, the access to this type of strategies is really limited with respect to the years that we are living now. So this means that in future, the situation goes better. And this for sure. But we need also to think that the complexity of these type of strategies are growing and growing up. No, in example, if you think to ovarian cancer, 5 years ago, there isn't any biomarkers from predicting molecular pathology side in ovarian cancer. But today, we have one of the most complex biomarkers in ovarian cancers, that is the analysis of HR status, so the so-called HRD in this type of patients. So the deficiency in the supervision of the repair strategies that our cells have. To analyze these type of biomarkers, we need to use not only NGS, but we need to use comprehensive genome profiling. So this means that complexity of these strategies is higher than a target NGS sequencing-based approach in which you are analyzing few genes. So the main point, another time, is not the analytical side of the strategies, but is that we need to prepare a group larger of professionals that are able to decodify this complexity, because we can have a bottleneck in the access to this type of human resource, not in the type of machine or the panel that we need. So we need to have fully validated analytical strategies, but for sure you need to have people that are able to manage these fully validated analytical strategies.
Amazing. And so maybe just to end along those lines, what would you say directly to the next generation of scientists and clinicians? What would you tell them about the role they can play in precision medicine to save lives?
Represents the art of all this process, because not only in the future, but already in the present, you need to have this type of people that are prepared in several landscapes. So in medicine and surgery, in biotechnologies, in engineering, because you need to have an interface with several professionals that are speaking several different languages. So the new generation of molecular pathologists need to be really contaminated in this sense, because you need to speak with bioinformaticians, with engineering, with your colleagues that are in charge of the morphopathology analysis. So it's really fascinating, but the key word is education. So we need to design and redesign, in some cases, the educational path of the new generation of scientists, to making in place to decodify all the complexity that we have discussed, because after that you have completed the next generation sequencing run, starts your job, because this is the time in which you need to decodify from the analytics to the clinics the power of the data that you have in your hands. And you can have some other supports coming from these really fascinating tools like as machine learning, deep learning, artificial intelligence for sure, but you need to manage all these tools and to apply these tools to decodify the clinical complexity that you need.
Thank you so much, Umberto. Definitively a field where there is a future. We need a lot of smart and clever people. Thank you for your time today, Umberto. Thank you for your impact.
My pleasure.
If we want to scale data-driven medicine, we need pioneers like you who speak both science and truth. You've done that today. This is Decoded. I'm Georgi Camblon. More episodes are coming with the voices shaping the next chapter of healthcare. Subscribe, share, and if you're working to bring better care to more people, keep going. See you next time.